Hey class, welcome to chapter 9 lecture. I'm going to divide this lecture up into 2 or 3 different videos. I think the videos are just taking too long to upload to YouTube, and unfortunately they're timing out, so it's taking me forever to get these up and online. So let's go ahead and dive right into the first half of this lecture. So this lecture, or this chapter, really is all about trying to predict the traits that the phenotypic traits that offspring of parents will have. And so we call this field classical genetics. So let's review really quick what we learned. What is that noise? Let's review really quick the concepts. Oh, I just realized I'm in a weird spot. Let me move myself down here. Right in the middle of the screen. Okay. All right. So let's review really quick how meiosis occurs in the production of our sex cells and the production of gametes, and remind yourself how what is true, which of the following are true and which is not true about gametes that are produced from one meiotic event. So go ahead and pause the video, try to look through your notes if you can, and see if you can select the correct answer. So let's review each of these points, because I think this is actually a nice review to remember what our sex cells look like. So when we talk about sex cells, of course, we're talking about our gametes. In a female of the species, we're talking about an ovum or an egg. In the male species, we're talking, of course, about sperm. And these sex cells, these gametes, are very different because they are haploid as opposed to diploid, which all the rest of our cells, our skin cells, our muscle cells, our nerve cells, and so on, they're all diploid. They contain 2 copies of, I shouldn't say copies, 2 homologous chromosomes, one from mom and one from dad. However, our gametes only contain half the normal number of chromosomes as our autosomes, our non-sex cells. So we know that the process of sexual reproduction, the whole point of it really is to introduce genetic variation. So the answer, the letter A answer, they're genetically identical to one another, is not true. We know that there are a couple of events in meiosis, crossing over, independent assortment, that causes, independent assortment, that cause genes to vary from one gamete to another. So we know that A is not true. We know, of course, that gametes are not genetically identical to one another. Hopefully you've already selected or recognized that B is the correct answer, but let's go ahead and continue on to the other options and talk about why they're not true. So if we look at question number C, or answer C, question number C, how many more things can I get wrong in that statement? If we look at option C, answer C, we know that one cell would produce, at the end of meiosis, will produce 4 daughter cells. And all 4 of those cells are going to be different from one another. So this is, again, a very important aspect of meiosis, in that we're producing 4 genetically variable cells, not only from one another, but from the parent cell. So we know that they're not genetically identical. And of course, I just addressed letter D as well. We know that they're not genetically identical, and therefore we know that E can't be true also, that they can't have the same mutations. So we know that A, C, D, and E are really all variations of the same kind of answer, in that they're not the same as one another. And this is the exact opposite of mitosis. Remember, when we have a mitotic division, so when our skin cells divide or our cardiac muscle tissue or muscle cells divide, we want those 2 cells to be exactly the same as one another. So in mitosis, the important part of mitosis is, of course, that those 2 daughter cells are genetically identical to each other and to the parent cell, because we don't want variation happening. When we have variation happening in mitosis, we have big problems. We probably have cancer or some other extreme genetic problem in those cells if mitosis is causing genetic variation. In meiosis, it's really the exact opposite. We want genetic variation because we want, if we have lots and lots of offspring, imagine the prolific offspring that plants and other sexually reproducing animals have. They have hundreds and sometimes thousands of offspring in their reproductive lifespan. And they want as much variation in those offspring as possible so that they can adapt to their environment. And that's what we're going to talk about in the next unit, in unit 4. All right, so the answer, of course, is that they have the same chromosome number. All gametes, sorry, I think I have to hiccup. all gametes have the same chromosome number, which is 1N, which of course is 23. We're going to have 22 autosomal chromosomes and 1 sex chromosome. And the reason that, of course, our gametes have half the normal number of chromosomes is that that gamete is going to fuse with its counterpart. If we're talking about an egg, it's going to fuse with a sperm to form that zygote. And now our zygote has that normal, our diploid number, 2N number of chromosomes. So this slide actually summarizes pretty much everything that we'll be talking about in Unit 3. Modern genetics is really talking about how alleles work together. So right here, these are our chromosomes. We have 2 homologous chromosomes, one from dad, one from mom. Our alleles are found on a specific location or locus on each of those chromosomes. And that gene, of course, codes for nucleic acid, a very specific nucleic acid sequence that eventually will give rise to a protein. In this case, this protein has an enzymatic function, and that particular enzyme causes the purple color to develop in this case, a pea plant, the flower of a pea plant. And so if you can follow this flow of information, this is really what we're talking about in Unit 3. We talked about how chromosomes are organized and distributed in Chapter 8. We're going to talk about what alleles are and how they eventually lead to this pea plant color, the purple pea plant color or white or whatever it might be. And then in Chapter 10, we're going to talk about how DNA actually does that. How does DNA turn into a protein, an enzyme? So let's go ahead and continue on with our genetics. So the idea of genetics has actually been around for thousands of years, but of course there were all different kinds of hypotheses on how we inherited traits from our parents. Everyone's known from for ages now that of course we inherit traits from our parents, but no one really knew the exact mechanism until recently. And really, to be fair, we're still working that out. There are still very interesting discoveries being made about inheritance and the way that inheritance is the exact mechanism of inheritance. So it's a really cool field to be in if you like stats, because it's a really math heavy field. So early on, there was this idea of pangenesis and another hypothesis called the blending hypothesis. And pangenesis really meant that it was a hypothesis, of course, that made sense at the time because we didn't have molecular biology and we didn't have biotechnology at the time to really analyze genes and chromosomes. We didn't even know what genes and chromosomes were at that time. But pangenesis was really a theory that said that as you grow and live your life, you're going to develop certain traits, and those traits will travel from your body into either your eggs or your sperm, and changes that occur in your body over that time will affect that egg or that sperm. And so what's really interesting about this hypothesis is that there are some really interesting epigenetic changes or epigenetic discoveries that have been made in the last 10 years that are actually shedding some light that this idea that what happens in our lifetime can affect our offspring is coming around again. So there are some really interesting studies that show that the chemistry of the womb, for example, is turning certain genes off and on in a developing embryo. And they got the first hint of this when they cloned a cat. I don't know why I'm talking about this. I'm lonely. I'm in quarantine. I have no one to talk to. But they got this first idea that there was something more to the story when they cloned a cat, and they cloned a bunch of cat embryos, and they put it, of course, in a mama cat for the babies to develop, for the embryos to develop. And when the cat gave birth, all the babies were different. All the kittens had different colored fur and had different colorings. And of course, when this first happened, they thought they screwed up. They thought they had made some mistake in the cloning process. But when they went to check their work, they realized, no, these are true clones of one another. And somehow, one way or another, some of these genes are getting turned off and on during development. So it's a really fascinating area and a newly developing, the idea of this epigenetic change in gene and inheritance is a very new idea. So, and it's also, as you can see, a very old idea as well. The other hypothesis, of course, is called the blending hypothesis. And this is the idea that there were just basically the egg and the sperm are some chemicals. And when they fused, they mixed their chemicals together, much like we're mixing paint together. And those traits kind of had like this 50-50 mix of both traits of the parents. And again, another fair hypothesis to make in the absence of molecular biology tools, but we know now, of course, that that's not true, that we inherit discrete packages of information called genes, and we inherit one from our mom and one from our dad, and that those two traits aren't going to necessarily blend together, but one is going to be dominant. In a typical model, one will be dominant to the other. So no talk of modern genetics can really be had without mentioning Gregor Mendel. He's considered the father of modern genetics. This is, where is he? Which one is he? Let me see. Oh yeah, he's right here. So and he's holding a plant. That makes sense. So he's right here, and he's holding some type of plant, it looks like, and he was a brilliant mathematician, a brilliant statistician, and his true love was physics, but he couldn't afford to go to university and stay in university at the time. This was, almost 200 years ago in the I guess he would have been making this decision in around the 1850s, and he couldn't, his parents were both farmers, and he couldn't find a sponsor to send him to university, and he didn't come from a family of means, and so he couldn't afford to stay in university and become a teacher like he wanted to. So he ended up joining a monastery, not because he was religious really, but because he basically wanted to be left alone to do his experiments, and he got it. He was left alone, and he was able to do these really prolific experiments on all different kinds of plants and publish his findings, which of course were then ignored for about 50 years until the early 20th century, and everyone finally realized what amazing work he did in terms of figuring out what genes were and how they actually functioned. So Mendel's work focused on the inheritance patterns of characters. And so characters is like, you can kind of think of character like a category. It's a feature that can vary among individuals. So for example, he studied, of course, Mendel had his famous experiment based on pea plants. So everyone knows what a sweet pea is, or you have the really great sugar snap peas that you can buy at the farmer's market. And they make these really beautiful flowers. Some people grow them just for their ornamental value. But let's talk about the different characters that that you can look at when you're looking at a pea plant, for example. So you can look at flower color, you can look at seed color. They can either be yellow or green. You can also look at the seed shape itself. It can be either round or wrinkled, and so on. So all of these are considered characters. While on the other hand, we have the idea of a trait. And so the trait is the specific variation of that particular character. So the fact that flower color in this case had only two options. It could either be purple or white. The color of that flower is the trait itself. So seed color can be yellow or green. Seed shape, again, can be round or wrinkled. So we want to differentiate between traits and character. So I think I have a little question. Yeah. So ask yourself, which of the following are traits? So hopefully you could recognize right away that the first two, hair color and eye color, aren't traits, they're characters. They're a category in which we can have variation. So hair color, we can have brown hair, we can have blonde hair, we can have black hair, and so on. We can have red hair. Eye color, brown eyes, blue eyes, green eyes, and so on. So hair color and eye color are both characters, whereas brown eyes, of course, would be a trait within that character. It's a variation on the character of eye color. Type A blood would also be a trait. So blood type would be the character, and blood, the fact that you're type A or type B or type O, whatever it might be, is the trait itself. On the other hand, 160 centimeters tall would be a trait, and of course the category would be height. You can measure your height, and the how tall or short you are would be considered your trait for that category or that character. So there was really, there we go. when so as a person who has done science and actually done research science, one of the most important things that you can do when you're designing an experiment is pick a good model. And so when I think about Mendel, I think about how lucky he got. Maybe he tried lots and lots of plants, but he really chose an ideal model for studying genetics, and he chose the pea plant. And he chose the pea plant for several reasons, and they're all just perfectly suited to do the type of genetic experiment that he had. First, they had very distinguishable features. You can easily see the color of the flower, for example. He wasn't studying something that's very hard to see, or that maybe you had to see with a like for example, he couldn't study blood type because at the time, of course, no one even knew what blood type was, and they couldn't distinguish between type A blood and type B blood. So that's not a good model in 1860. Where did he live? In Hungary, I think. He was part of the Austro-Hungarian Empire. This is, in the 1860s and so on. So he needed a model that had very easily distinguishable traits: purple flower versus white flower, yellow seed versus green seed, and so on. Another nice thing about pea plants is that they can self-fertilize. In other words, he can use the same plant to pollinate itself. And this allowed him to breed plants that were true. In other words, all of the plants that he started with in the parent generation were homozygous. And that will come up. I'll talk about why that's important later. And while those plants were able to be self-pollinated, he could also quite easily cross-pollinate them. So if you've ever done this, if any of you are amateur gardeners, right, you have your stamen and your pistil, and you can basically either pollinate the flower itself, or you can cut off the pistil so that it's unfertilizable, and you can do this yourself with like a little brush, and you can fertilize plants to try to create the characteristics that you want in your own garden. And then the last two really have to do with the efficiency with which he was able to carry out this experiment. Pea plants grow really, really fast. They have a fairly short life cycle, and they produce lots and lots of offspring. So this allowed him to basically do a lot of repeated experiments in a very short amount of time and get lots and lots of really good data so that he could run the numbers and do the math and create those data tables to analyze what he was trying to investigate. So he ended up looking at seven different characters. Seven? 1, 2, 3, 4, 5. Yeah, 7. He ended up looking at 7 different characters. Each of these characters had 2 traits. So again, a really well set up experiment. He had lots of possibilities to choose from: flower color, flower position, the different colors of the seeds, how the pods looked, and so on. And each of these characteristics had either one option or another, which is it's a really nice model again to study. For example, if you look at the stem length, they were either tall or short. You didn't have like tall, short, and middle, for example, or you didn't have green seed pods and yellow seed pods and then something that was somewhere in the middle that was kind of like some combination of those two colors. So he had a really nice model set up here where he had 2 different traits for each particular character that he wanted to look at. So here you can see, here's what I was talking about in terms of the technique that he used. And I want to just very briefly, in order to propagate these pea plants, he could cut off the, I believe this is called the pistil, I forget, basically the female part of the flower, and then he could take the stamen, basically the pollen from one flower, and paint it onto the pistil. That carapace? What is the name of that thing? Ugh, I should look it up in my book. I'm too lazy. The female part of the other flower. And he painted the pollen on there, of course, to fertilize this flower. And of course, this is their offspring, these seeds that you get out of the pod. And then these seeds would make that next generation. So let's talk about some vocabulary here alongside of the technique. So true breeding, I mentioned this term already. True breeding means that these plants have been self-fertilized and that the offspring will always look like the parent. In other words, if you continue to self-fertilize these plants, you will always get the same plant again and again and again. In other words, they breed true. There will be no variation in those traits. And so that was a very important part of his experiment because he didn't want variation to begin with in that parental generation. On the other hand, a hybrid would be when we have cross-pollination occurring or cross-fertilization occur, where we have an offspring of two different parents that have different traits. So we'd call that plant a hybrid plant. And when we're talking about generations, it's kind of weird to think about generations in plants, but whenever we begin one of these experiments, our original true breeding generation is going to be called the parental generation P1, or just P. And then their first offspring, that first filial generation, is going to be called F1. And then, of course, if we have a generation after that, it would be F2, and then F3, and so on. So we only have one parental generation, and then we have our first generation after that would be F1, the next F2, and so on. So let's look at what he did in his first experiment. So let's just look at one of these characters. Let's look at flower color and the different traits. Of course, remember, we can either have a purple flower or a white flower. So in our parental generation, he took a purple flower and crossed it with a white flower to get the F1 generation. So here we have our first hybrid flower. And no matter how many times he did this, he would only get purple flowers. So he never had two flowers, a purple and a white flower from his parental generation that ever produced a white flower. So he asked himself, is there any possibility, did the purple trait, did it just somehow like get rid of the white? Is the white trait gone? Is the ability that inheritable trait that codes for or brings about a white color, is it just gone forever? So he bred these flowers together. So he did another cross, it could be either a self or a cross-pollination, to create that F2 generation to see if they would be all purple. And you would assume that because all of the F1 generation were purple, that all of the F2 generation would be purple as well. And he was surprised. He was surprised to see that that's in fact not the case. There are white flowers, and there's actually plenty of white flowers that pop up in that F2 generation. And even more importantly, they always showed up within the same ratio. In other words, all of the F2 generation, so 75%, right? 3/4 of the flowers would be, 3/4 of the flowers would be purple, and 25% or 1/4 of the flowers would be white. And so he consistently got this ratio of purple to white flowers. So that was kind of his first hint that he saw that this wasn't a blending theory, right? The pangenesis or blending theory couldn't be true. So there had to be something else going on here. We know, of course, that if we blend, right, again, just to kind of address the blending hypothesis, if we were to blend or if we were to cross a purple flower and a white flower, we basically should see a bunch of lilac colored, right, like kind of lavender colored flowers. And we don't see that. We see this very specific ratio of 3:1 of purple to white. And not only did he see that in his flower color, every single trait that he tested for, seed color, seed shape, pod color, pod shape, and so on, all of them ended up having the same ratio, that 3:1 ratio. So he knew that it couldn't be just a simple blending of traits between mom and dad. So let's go back and remind ourselves how this works, right? Because we all know, of course, that we get something from mom and something from dad, but again, at that time, we didn't know what it was. And we now know, of course, that mom and dad each give us a chromosome, one of their chromosomes, and those chromosomes have specific alleles on them, and those alleles are different from one another. So for example, our dad could give us the allele for purple flowers, while our mom could give us the allele that codes for white flowers. And this was this was his big advance, that he said there has to be some unit of inheritance, and that unit of inheritance codes for a particular kind of trait. And further, that unit can't be changed in any way. It can't be blended or kind of modified in any way. It's intact. And this led him to kind of conclude that the way that genes vary, or the way that we get genetic variation, is through these alternative versions of alleles. And these so these alleles are variations on a gene that are located in the same place on homologous chromosomes, right? We consider these two chromosomes, one from mom and one from dad, a homologous pair because they code for the same, or they contain the same genes. They contain different alleles of the same gene. And the way that you can kind of think of a gene, I like I think this says it in your book, but a gene is kind of like a playing card, right? If you have the Queen of Hearts, for example, you can shuffle your deck of cards as many times as you want to produce variation, but the Queen of Hearts itself will never change. The Queen of Hearts will always remain. It won't suddenly become the Queen of Clovers or the Queen of, some other, it won't modify the heart into some other card suit. Another one of Mendel's conclusions was that we have to, or any organism that inherits or reproduces sexually has to have the inheritance of two different alleles. So for every character, there has to be two alleles and one from each parent. And of course, we see that here, right? Here's our first allele from dad for the purple flower. Here's our other allele for white flower color from mom. Here's our chromosomes. I thought this would be kind of a fun picture to include. This is called a karyotype. I left this out actually of the chapter 8 lecture, so I thought I'd just show it here. Here we can see all of our homologous chromosomes, one from mom, one from dad. Of course, we don't know which one is which. And here you can see that we have 22 homologous chromosomes, 22 homologous autosomal chromosomes. Remember that the autosomal chromosomes are basically our non-sex chromosomes. And you know that this is for a male offspring, one, because it says it in the title, but also because we see that X and Y chromosome for our sex chromosomes. Here, right? So we always get that X chromosome from mom. And in this case, the dad, the sperm, donated a Y chromosome. So this offspring will be male. So Mendel concluded, I'm going to try to, let me see. Oh, okay. Let me finish up these last couple slides. I want to try to keep this under a half hour. So his next conclusion was the law of dominance. So not only do we get two alleles, again, one from our female donor, one from our male donor, but these two alleles often differ. And when they differ, there's going to be one that's dominant to the other. So we call the dominant allele the one that produces the appearance. So whatever this organism's appearance is would be considered the dominant allele, in this case, of course, purple flower. And the recessive allele has no noticeable effect on the phenotype or the appearance of the organism, in this case, of course, the flower. So this idea of a dominant and recessive allele, again, as opposed to, think about blending two kinds of paint. If we have blue paint and yellow paint and we blend them together, we're going to create green paint, of course. And that's, Mendel is saying this is not what's happening in genetics. When we combine two alleles, one allele is going to be shown, one is going to affect the appearance of that organism or the function of that organism, and that's our dominant allele, and one isn't, and that's going to be our recessive allele. So let's finish this lecture with this little kind of review question. If you've done the lab for this section already, this hopefully will be pretty easy for you. A fish inherits two alleles, one for blue fins and one for red fins. The fish has blue fins. Which allele is dominant? So which allele is leading to the physical appearance? Of course, we know that it's the blue fin, because he has two alleles. One is codes for blue fins, one for red. But the blue fin is the one that he actually has. His phenotype or his physical appearance is blue finned. I don't know why I'm saying that it's a him. I guess the fish could be a woman. But let's ask ourselves, if the dominant, let's just use the letter F for our alleles. If the dominant allele is capital F and the recessive allele is lowercase f, this fish, what would the genotype of this fish be? course, the genotype is heterozygous because he has one of each. He has a capital F for the bluefin and a lowercase f for the redfin. And this fish would have a phenotype, of course, of blue fins because that's what is actually that's his appearance, is a blue-finned fish, which would lead us to having the correct answer of B. He has blue. The blue allele would be the dominant allele in this case. Okay, we're going to leave it off there, and I'll pick up with part 2 of this lecture, starting off with autosomal dominant traits.